Welcome, welcome. Thank you for all who are joining. Great to see your names appearing there. The big day has come. Thank you so much for joining us today for the debut webinar of CellarisBio. This will be a great program, and I'll present myself. My name is Brooke Minson. We'd like to say thank you so much for connecting today. And feel free to use your little buttons below. You can raise your hand, let me know that you can hear me okay. Click your little raise hand button and we'll know that you are here and present. Thank you so much. The talented professionals here of CellarisBio have prepared a fascinating and informative presentation, especially for you. So I'd like to introduce our two speakers. First of all, Elmar Nurmamedov. Dr. Nurmamedov is a co-founder at CellarisBio. He is an entrepreneur and scientist with PhD in molecular biophysics. Trained at Harvard Medical School and Scripps Research Institute, later principal investigator at St. John's Cancer Institute, his lab focused on in vitro pharmacology and early drug discovery. Elmar has contributed to development of novel cell target engagement methods. Dr. Nurmamedov has held positions of Director of Business Development at various companies, and Elmar holds patents for small molecular therapeutics and is inventor of several novel drug screening technologies. Round of applause for Elmar Nurmamedov. Now I'd like to present Dr. Ivan Babik. Dr. Babik is a co-founder at CellarisBio. He is an accomplished cancer researcher with a PhD in biochemistry and molecular biology from the University of Calgary. He has over 15 years of experience in drug discovery and has been actively involved in preclinical and clinical drug development. Prior to founding CellarisBio, Dr. Babik was assistant professor at St. John's Cancer Institute, where his lab focused on identifying drugs that would impact tumor metabolism. Dr. Babik holds patents for small molecule therapeutics and is inventor of several novel drug screening technologies. So there we have our two speakers, co-founders of CellarisBio, who you will meet today. Please get comfortable. And just a few details about the presentation today. You can expect it to be about 30 to 40 minutes. But don't get lost. Don't stray from your screen because it will be interactive. There will be several poll questions that we would very much appreciate your votes. So be ready to throw in your votes there. And also at the bottom of your screen, you should see a small button that says Q&A. Please feel free to activate that and throw in any questions you may have throughout the program. At the end of the presentation, there will be an opportunity to receive answers to your questions that the speakers choose. They will be the only ones to see your questions. So to begin the presentation, we'd like to direct our attention to Dr. Elmar. Go ahead, you have our full attention.
Brooke, thank you so much for that introduction. So I hope we can cover some aspects of cellular target engagement and what it does for drug discovery and why it's important these days. So I will go ahead and open up with this slide, target selection and why it's important in drug discovery altogether. As you well know, in the drug discovery field, things start with the selection of the right target. So selecting the right target means that you select the disease indication, you are setting pace for your drug discovery programs, and selecting the target also sets the pace for the economics of your drug discovery. So you could choose within multiple drug indications, could be cancer, could be neurological diseases or metabolic diseases. So, this is an image from the old Nature magazine. It's a smart target shopping. It's actual today as well as you select and start the path, the journey towards championing your drug discovery program. Many aspects are important in selecting the target, as we know that there are more than 30,000 human genes. There are only 3,000 of those genes are disease modifying genes, and out of that today we know that there are only about 1,000 drug targets that could be tractable that in a disease-modifying way. And with all these advances in drug discovery, methodologies, in the pharmaceutical science, we come to agree that more than 80% of these drug targets are still undruggable. So there are various ways to approach these targets. The field is moving forward, and there are challenges ahead. But this is the big picture in target selection and drug discovery. So just wanted to give you a very short, high-level overview about the conventional biophysical methods. In the earlier days, the field of protein science, protein characterization, there have been a number of biophysical methods. These are only 4 methods that I'm showing here that have really benefited the drug discovery, the protein field. So this is surface plasmon resonance, SPR, it's ITC, MST, and TSA. So essentially, they measure, they're based on various principles of discovery. They measure optical changes in the signal. They measure the heat exchange, they measure the motion, and they measure the shift in the melting temperatures. And there are various benefits to these temperatures, like they can measure the kinetic constants, the on and off rates. They can measure the enthalpy and the heat released, and also shift in the melting temperatures. Throughput-wise, these could be low throughput, like this ITC, or could be a high throughput like surface plasmon resonance. What is really important is really that all of them require a purified, soluble, recombinant protein. So whatever target you are working with, you will have to express and purify the target in whatever system, like E. coli, yeast, or in some other models, and have the system, the protein really purified and soluble and well behaving before you can even start using these methods. This could be a disadvantage. It means that you have to take the protein outside of its cellular context and really introduce some level of artificiality before you can really test those targets in your systems. Sensitivity-wise, these assays can go between the picomolar or up into the micromolar, millimolar level. And for some of these methods, we'll have to immobilize the protein on the surface of your reaction, and you may need to label the target or your ligand. So there are various ways. The conventional biophysics have done protein analysis, but most of them have relied on the purified protein, which is a major shortcoming. I would like to transition from here to the modern challenges of the modern drug discovery. So we know that there are more than 20 different methods in early drug discovery that the current drug discovery field is using, and this disrupts the cell biology. So the essential cell biology is leaked out because of this multiple step process. There are difficult or challenging targets, more than 600 of them out there, which require a non-conventional approaches, which require the cell biology to be present at all stages of that drug discovery. And there are more than 9,000 undiscovered therapies out there, which are hiding behind these challenging drug targets. So if we find new ways to approach and tackle these difficult targets, we have a better chance at these undiscovered therapies. And this means more economic value. This means more successful drug discovery programs. There are challenging targets and modalities in current drug discovery. So some challenging drug target families would include transcription factors, would be enzymes, would be membranes, ion channels, regulatory proteins, and protein-protein interactions. What makes a target challenging is, by definition, is when a target cannot function outside of its natural or cellular context. For example, take a transcription factor. It doesn't have a defined structure outside of the cell. It's pretty much intrinsically disordered. So the conventional biophysical tools may not function or serve well for these type of targets. Membrane proteins are similar. and other protein-protein interactions are, a similar situation where you cannot recapitulate their biology outside of the cell. And on the top of this challenge, we have now multiple therapeutic modalities, such as small molecules, active site binders, allosteric binders, and more modern tools such as degraders, molecular glues, peptides, and antibodies. Now, when you combine the challenge of these difficult targets with the challenge of these novel therapeutic modalities, you need a new system where, the cell biology would be present and your target of interest would have its structure and function all in one place. Cell target engagement really accelerates and de-risks drug discovery. As you may know, the current drug discovery can be very sophisticated. So on the left-hand side, you have your genomics and proteomics tools feeding information into your drug discovery platform. This could be, synthetic lethality screens. These could be various bioinformatical analysis. And once you have that target of interest in sight, you would do, the target ID, and you would develop assays around that target. The second stage would be the discovery stage, where you would screen for small molecules, either using conventional plated libraries or DNA encoded libraries, if you will. And once you have the candidates, you would go into hit to lead optimization and define and assign your lead compound, after which it would still need to be optimized and would go into preclinical studies. And hopefully from there, it would go into clinical trials. So the odds are stacked against you, and this is a very expensive process. In the end, what really matters is the question, is your drug really therapeutic? Does your drug engage its target inside of the cell, not outside of the cell under artificial conditions, but directly in the site? So the sooner you answer these questions, the better are your chances to making your drug discovery program a success in the clinic. So in order to beat the high failure risk in the clinic, it's important to address these questions early on so that you have a better chance to making this drug discovery program a success. So altogether, cell target engagement unifies the drug discovery process. It eliminates some of these artificial or otherwise redundant techniques that you may not necessarily use if you have the cellular target engagement. It preserves the cell biology. You may not need to express and purify the recombinant version of the protein anymore. You just may need to work directly with the cell of interest and preserve the important cell biology and preserve the disease relevance of your cellular model. So cell target engagement also streamlines your discovery. It adds to the speed and efficiency of your drug discovery processes, and also takes away some of the risks related to that discovery process. And altogether, it improves the future clinical success of your drug discovery program by asking some of the very important questions early on. So altogether, CellarisBio's cell target engagement platform sits right between the steps of hit discovery and hit to lead optimization, where we are able to really bring our technology forward and work with the target of interest and make sure that the discovery process, the validation of your drug candidates are done directly in the cell by eliminating the unnecessary risks related to it. I want to mention you a few things about the conventional cell target engagement and the principles of cell target engagement altogether. A little biophysical knowledge here is essentially, the cell is it's an environment, it's an ecosystem for thousands of different proteins living in their natural environment, and every protein has its biophysical footprint. So essentially, if you subject a protein to a thermal melting to a heat stress, the protein will essentially melt, and there is that sigmoidal curve that goes from high signal to a transition and essentially to a low signal. If we can identify the thermal melting profile of a target protein of interest, essentially, then we can interrogate small molecules to it. So on the right side, you will see a transition state of a protein as it binds to a ligand. You will see a protein that is structured when a ligand is bound to it, to protein of interest. It will rescue the target protein from denaturation, which would lead to that delta T, to a temperature shift by which your protein has been stabilized by binding to a ligand of interest. This could be a small molecule, this could be a degrader, this could be a peptide that will selectively bind to your protein of interest and stabilize it. And this is essentially the principle of cell target engagement. There are various advantages to it. Cell target engagement is done under physiological conditions. It relies on thermal melting profile of your target of interest. There are minimal modifications required for the target, so you don't need to excessively make it recombinant. You don't need to take it outside of the cellular context. No modification or labeling of the compound is necessary, and the system can be disease relevant, and this opens up floodgates to multiple applications such as discovery, validation, and even analysis of drug discovery. So cell target engagement by itself could be an end-to-end solution to early drug discovery. So there is a conventional first generation of cell target engagement, which uses immunoblotting for detection purposes. And this technology here really opened up a new paradigm in cell target engagement. So the conventional way would be to measure the target of interest directly within its natural context without the endogenous version of that target protein directly in the cell. There are two different ways to approach this. Either use the intact cells or use the non-denaturing lysates of those cells. This system would then treat the cells with the ligands in dose-dependent manner, importantly subject the samples to heat stress, and then lyse. And another important step is the centrifugation step, which would separate the soluble versus the non-soluble version of that target protein. So you would only work with the soluble fraction of your samples, which could lead then to Western blotting detection by immunoblotting, or you could do mass spectroscopy analysis. Now, this is a great technique, and this has been a trailblazer when the field started. There are advantages to it. No modification of the target is necessary. No modification of the compound is necessary. There are a number of disadvantages. This is based on immunoblotting. You would need to have good antibodies for the target for this to work. And this could be low throughput, but it functions well. It could be laborious because of immunoblotting and the centrifugation steps. And this technique also has a disadvantage of low sensitivity. So a compound that is nanomolar affinity binder could appear as if to be a micromolar binder because that sensitivity might be lost during this process and could be limited sometimes to certain well-behaving target classes. And this is one example of immunoblotting-based cell target engagement.
Elmar, I
Do you have a
hate to
question for me?
interrupt you. I have a
Okay, please.
question for you. Are you ready to open up a poll for the audience to get their input up till now?
Yes, let's do that.
Let's
Okay,
do
so
that. Get ready to notice there's going to pop up on your screen a question, and we'd really appreciate to hear your feedback. You can select a choice from the list. It will be anonymous. So take your time to read the question there. To what extent are you familiar with cell target engagement? So choose one of the four options below. You use it often. I seldom use it. I never use it. I am not familiar with it. Please select your answer and give us a little raise hand icon like that when you are ready. Thank you so much for your participation. We really appreciate it.
All right, should
When,
I continue now?
go ahead, Elmar, whenever you're ready.
Okay, so this is one example here of immunoblotting-based cell target engagement. Now we have a compound here, compound 6125, that is supposed to bind to PLK1, and you can see that as it binds to PLK1 under that temperature stress, at higher elevated temperatures, there is more and more stabilization of PLK1, and we can see that as an intensity in the immunoblotting band. Great, works well. And you can also actually play on the selectivity, and we can see that PLK3, which is an analog of that target, does not bind to this compound. And you can plot the data as a graph, and then you can extract the EC50, which is 0.65 for the real target and much higher for the other target, except that we know this compound is more sensitive when we measure as using other non-conventional techniques. So this immunoblotting based approach is great, but the sensitivity is somewhat compromised. So now there is a second generation of cell target engagement platforms. This is a technology that is based on enzyme complementation. And with the principle of this technology is that there is an enzyme where its small subunit is clonally attached to your target of interest, and the larger subunit of that same enzyme, together with the detection reagents, is added to your solution. And when the two complements find each other, essentially it gives rise to luminescent signal, which is proportional to the amount of stabilization of your target protein by your ligand. And you would see curves like this. Essentially, you can measure the thermal melting profile with or without the compound. Or you can also measure the EC50s, the potency of cell target engagement with various compounds. This could be scalable, this could be sensitive approach, and there are advantages to it. It's sensitive, scalable. It can be used for both discovery, validation. This could be automated if it's working well in your hands, and the data could be rich enough for other potential applications. These advantages include minimal modification or labeling of the target might be necessary, can be limited to certain classes of targets, like, where the tag and the spacer would be functional. And sometimes, or many times, this approach would suffer from poor time resolution because it's based on luminescence. And in many applications, this is used for measuring of endpoint signal instead of that, time result signal. So there are great sides to this, but there are limitations to this second generation technology. Now, I want to switch now to gears to the microtag cell target engagement, which represents the third generation of cell target engagement technology. So as you have seen, the field of cell target engagement technology has evolved over the past decade, since 2023. So on the right side, you see the immunoblotting-based technology, which is essentially sensitive. It's a start of this field, but then there are aspects to it that are not there, like scalability, like the target agnostic feature, the real-time feature, and ability to use with DNA-encoded libraries and AI integration. Essentially, those features are not available for the first generation immunoblotting-based technology. Now, the second generation technology, which is based on luminescence, on enzyme complementation, is sensitive, and it's scalable as well. And there are very good publications and there are good applications to it. There are still disadvantages because it could be limited to certain well-behaving targets. The real-time aspect to it is not available, or it's poorly available, poorly developed. The DNA encoded libraries aspect to it is not there, and AI integration is also not there. So this makes the second generation technology limited for certain uses. Now, CellarisBio's Third generation cell target engagement technology is based on fluorescence. Now, there is a new chemistry behind it. Because of these features, because how the technology is built, it could be sensitive, it can be scalable, it's target agnostic, you can use for various target classes. It can be real time if you need to understand and measure the mechanistic aspects of your drug target pairs. You can complement this technology for discovery using conventional or DNA-encoded libraries, and you can complement the AI. You can integrate this technology with AI as well. Just to delve deeper into comparison between fluorescence-based and luminescence-based cell target engagement technologies. Now, the luminescence-based could use the tag size, which can go from 12 amino acids to 42 amino acids, whereas in CellarisBio's case, it can be 12 to 15 amino acids. CellarisBio uses fluorescence signal, which gives you the ability to use with unlimited target classes. There is no need to use any tracer. The signal could be in real time. If that is a requirement for you, you can see as the reaction progresses from time 0 to a later time point, and sensitivity can pick up engagements between targets and the ligands from the picomolar all the way to the micromolar level. So it's very much sensitive, very much scalable system that can make a huge difference for early drug discovery programs.
Are you ready to launch our second question, Elmar? Sorry
Let's
to
do that.
interrupt.
Okay, let's do that. So question
Let's hear
2.
what the audience says. Get ready to click your answer. Send us your feedback as soon as it pops up on your screen. There we go. What type of cell target engagement method do you use? And you can choose multiple answers of the 4 that you see below. And like the last time, send us a little raised hand when you've plugged in your answer. Thank you so much for your participation. Whenever
Great.
you're ready, Elmar.
Yeah. So just to have a look on how this microtag system is designed, it's very important to understand that there is a 15 amino acid tag that you have to clonally attach to either N or C terminus of your target of interest, and this will generate for you a microtag, engineer a microtag reporter cell for that specific target. Could be a transiently expressed, could be a stable expression of that stably expressed reporter cell. Once you have that reporter cell functioning, you can treat it with your ligands of interest in whatever format, dose dependent or single dose. You would need to apply that temperature stress, which needs to be empirically determined. You would then detect the signal by adding the larger subunit, the tag binding protein, and the FRET substrate. Measuring of the signal could be done in various different equipments, which are readily available in many labs. This could be a real-time reader. This could be a real-time live cell imager, or could be a conventional plate that reads fluorescence signal. So it can give you various applications depending on your needs. And just to recap on the essential features, it uses 15 amino acid as a tag, which is clonally attached, works by enzyme complementation, interrogates the target by its thermal challenge, and uses fluorescence signal for readout. And please put your questions in the Q&A session so we can answer them after this. Just to show you the workflow with a microtag cell target engagement, essentially the first thing would be selecting your target of interest. What is it that you want to target? Could be any structural or functional protein class. The second one is you engineer the microtag reporter cells for that specific target. The third thing is then performing the assay, which could be hit discovery, hit validation, lead optimization, and then real-time analysis can really add a lot of speed and efficiency to your medicinal chemistry and early drug discovery efforts. And then it leads you to proceeding faster into preclinical studies with a lot more confidence. So just want to recap some of the important features of our cell target engagement platform. So we work routinely with difficult or challenging drug target families, which could be enzymes, transcription factors, membrane proteins, other difficult targets. We work with multiple therapeutic modalities here. These could be small molecules. These could be peptides, degraders, molecular glues, and antibodies. I have to say that more than 80% of our applications are in the small molecule field, but we do have a number of applications and pilot studies for the other therapeutic modalities as well. Before I hand it over to Ivan, I want to recap some of the important features of this technology. Target agnostic, as we have seen, enabled with DNA-encoded libraries if that is necessary for you. Sensitivity of this technology because of its new chemistry, scalability, the way the reporter systems are assembled and the way it can be used on a scalable, repeated basis. And then the real-time application, which could be important for your mechanistic aspects of your drug target pairs, and then the entire data that we can generate using this technology can be plugged into AI ML application to enrich your your medicinal chemistries, to speed it up and to generate more robust medicinal chemistry programs for you. Brooke, is there a transition point for us?
Absolutely. That was so interesting, Elmar. We have one question to launch to the audience before we pass to Ivan. Can you set that up for us? The third question. So get ready to throw in your feedback. For what purposes do you use cell target engagement? And you can also choose multiple answers here. Thank you so much for your participation up till now, and keep sending your questions also in the Q&A. Let us know when you've got your answers plugged in. Thank you with your hand. All right, Ivan, all ears for you.
Thanks for the introduction, Elmar. So I'll just present a few of the case studies that we have using the microtag cell target engagement system. The first one here is mitogen activated protein kinase, or MAPK1, also known as ERK2. So it's a well-characterized protein involved in several cancers, involved in resistance to cancer therapies. So one of the ways, so the way we start when we onboard a project is to understand a little bit about the biology of this target, because we have to make a decision to place the 15 amino acid tag either at the carboxy terminus or the N terminus. I noticed in the Q&A, there was a question, do you see any differences in tagging that same protein at the N and C terminus? We do. It depends on where the drug actually binds, and I'll mention something about that in the next case after MAPK1. So when we start with a particular project, we design or engineer the construct having that tag, again, either at the N or C terminus. We do a quality control test, first of all, just to make sure that the target expresses at the correct molecular weight. So make sure there are no degradation fragments, that the tag is being cleaved off. So we have immunoblotting with a microtag antibody to demonstrate expression. Following that, we then look at the fluorescent signal to make sure that that tag is amenable to enzyme complementation with its partner protein. And so you can see in panel B, there's an increase in fluorescent signal above untransfected control, suggesting that the tag is functional. So those are our quality control that we go through before we start testing any sort of drug binding to this particular target. Once we complete the quality control, the next step is to challenge this construct or this protein target to a thermal challenge to identify a thermal profile for this target, so we can identify the temperature at which 50% of that protein becomes denatured and aggregated. And so for MAP kinase, this is non-denaturing lysates that we prepared. 3 minutes heating from 44 degrees to 70 degrees. On the left-hand side, you can see a real-time curve. So this is using the real-time system by Thermo Fisher, the Quant Studio 3 real-time system. We see at about 20 minutes, we can take the data points from the 20-minute real-time curves and plot that against the temperature. And so we generate a thermal profile for MAP kinase with an identified temperature of aggregation of 54 degrees. So that's the temperature at which we will then choose to interrogate drug binding. So these are just some tests. This is a negative control compound, first of all, incubate with the lysates that were isolated from MAP kinase 1 expressing cells. Incubate for an hour, heat challenge, and this is the real-time curves for this negative control compound. When testing other compounds that are commercially available, well-characterized MAPK1 binding compounds. So this is SCH-772984. I believe it's a Merck compound. You can see that there's an increase in the amount of fluorescent signal as you increase the concentration of this compound. And then another positive control compound, this is AstraZeneca, AZD0364 compound, well-characterized commercial compound that's a MAPK1 inhibitor. You can see that separation of the signal. So fluorescence starts to increase as you increase the dose of this particular inhibitor. So taking a derivative of this data, plotting the inhibitor versus the fluorescence, plotting the concentration of inhibitor versus fluorescent signal generated, we can identify EC50 values for target engagement. So these are very potent compounds. The AZD compound 0.5 nanomolar is determined, the SCH compound 0.6 nanomolar, and the negative control compound, there's just no binding. So demonstrating that we can identify binding of these particular compounds as demonstrated by an increase in the stability of MAPK1 target, which results in an increase in the fluorescent signal. So another case is that we want to present here is MTH1. It's a protein that is involved in DNA repair. Well-characterized protein with a number of commercially available inhibitors, both well-binding inhibitors and negative control inhibitors that are actually enantiomers of the compounds that do bind. So again, when we start with a particular project, we need to know a little bit about the biology of this protein. Do we tag the carboxy terminus or the N-terminus? If there's a secretion signal at the N-terminus, we choose the carboxy terminus. So here, in this case, MTH1, it was tagged at the carboxy terminus, again, demonstrating its expression by immunoblotting with an anti-microtag antibody, showing that there's only one single product, showing that that tag is amenable for enzyme complementation, generates fluorescent signal. And then subsequent to this, we do a thermal profile to determine a temperature of aggregation. So these are real-time thermal profile using the Quant Studio 3 real-time system. And as we increase the temperature, you start to see a decrease in the fluorescent signal, resulting in a temperature of aggregation that we can derive of 55 degrees Celsius for MTH1. So using 55 degrees Celsius as our heat challenge temperature, testing two inhibitors. One is the well-known, well-characterized S-crizotinib, and its enantiomer, R-crizotinib, has been demonstrated not to bind to MTH1. So you can see the difference between the two inhibitors. So there's no binding of R-crizotinib as demonstrated by the graphs overlapping, whereas with S-crizotinib, with an increase in the amount of this inhibitor or the dose of this inhibitor, you start to see an increase in the fluorescent signal in the real-time curves. So we can use this data to identify an EC50 of target engagement. So if we take derivatives of this data, we can generate sigmoidal curves to, which I think is on the next slide. There we go. Generate sigmoidal curves whereby we can identify the EC50 of target engagement for S-crizotinib. R-crizotinib, there was just no binding. So just showing how sensitive the real-time data is, both for the MAP kinase and the MTH1, showing that when an inhibitor does bind, we get a nice increase in the fluorescent signal. Another case, very popular cancer target, KRAS, which is a driver of multiple cancers, mutated in colon cancers, lung tumors, lung cancers. So it's a very popular therapeutic target just because it's been shown to be a driver of cancer. So there's been a number of companies that have been trying to identify inhibitors of KRAS. So again, as we start with any project, we need to know a little bit about the biology of this particular protein. Here we've tagged KRAS at the amino terminus, demonstrate its expression, that it expresses really well in the cell lines of interest, that there's no degradation fragments. Looking at the fluorescent signal generated by the tag, we see a nice increase in fluorescent signals showing that the tag is functional. Then the next step would be, again, as demonstrated in the previous case examples, looking at a thermal challenge of the, in this particular case, it's KRAS G12C mutant version, tagged at the amino terminus, and these are lysates that were prepared from cells overexpressing this target. And so as we increase the temperature, you start to see a decrease in fluorescent signal, resulting in a temperature of aggregation that we can determine to be about 44 degrees in these lysates. So that's a temperature at which we now will challenge the target. So incubating KRAS G12C mutant with the Mirati Therapeutics compound, MRTX 849. I think its trade name, I guess, is adagrasib. You can see this differences in binding. So as you increase the dose of this inhibitor, you start to see an increase in fluorescent signal, which is a result of stabilization of this protein in a thermal challenge. Now, comparing that to the wild type KRAS form, there's just no increase in fluorescent signal, which is indicative of no binding of this particular compound. So what's interesting about this compound, it's an irreversible covalent inhibitor. So it only targets the KRAS G12C mutant, but will not affect the wild type form of KRAS. And so KRAS, the G12C mutant, is highly, the KRAS is mutated in a number of cancers, and G12C is a common mutation seen in a number of these cancers. So I presented to you a number of just 3 cases here, but the microtag target engagement strategy, or this system, is target agnostic. So we've performed a number of case studies that are not just kinases or well-behaving targets, but we've worked with a number of challenging targets, like transcription factors. For example, we have data with c-MYC. We've worked with a number of transmembrane proteins, such as EGFR, number of G protein coupled receptors. So it is target agnostic and amenable to a number of different types of targets.
Thank you so much, Ivan. Elmar, could you continue the presentation?
Yes, I just wanted to recap some of these points already made in the presentation. So this is the process for onboarding the targets into the microtag platform. So we always start with the reporter phase, which is essentially a small feasibility test, a quality control. So we want to know about the target, a little about biology, so we can prepare those reporter cells. And takes a month. Then we need to do those, simple tests to see how your target behaves in the microtag system. So then once successful, we go into the pilot phase. Pilot phase is a little bit of assay development. So we bring in a tool compound that might be available in your hands, or commercial available tool compounds to that particular target. And then we want to see how the target responds to ligands. Is there stabilization? What is the magnitude of that stabilization? And we want to see that pattern. It's an empirical process. We cannot guess it. We have to experimentally test that. And once we see that pilot test successful, then we can scale this up. And then scaling up could mean that you want to test more compounds. You can do a lot of validation, verification of your ligands discovered from your lab. You can do real-time analysis using, mechanistic aspects. Or you can actually choose to do a completely new de novo discovery, using plated conventional libraries or even DNA encoded libraries. So this is the process, and we have to go through this process before we can scale up and before the program could be successful. So if you have any targets that might be of interest to you, we would be happy to put that through a pilot test and see how it could be used and how the program could benefit from it.
Before we continue, Elmar, could we throw out the last question for the audience? Excellent. It's the last one. Throw in your answer. Are you interested in testing CellarisBio's cell target engagement technology? Please let us know. We're interested to know your answer. Thank you for letting us know that you participated in the poll. All right, Elmar.
Yeah, so a last slide here, really, to recap the three pillars of the microtag cell target engagement platform. So really, that one design of the cells of the system for that specific target opens up now various applications. You can always do validation. You can rank the compounds by potency. You can do target interrogation, test selectivity, or you can do analysis for a handful of most promising compounds. You can do real-time kinetics in the cell. You can do real-time live microscopy. And you might be also interested in a completely unique application, which is the discovery module of this platform. You could discover new chemical matter through pooled libraries or like DNA encoded libraries, or the conventional plated libraries. And depending on this, I think we could probably help you and guide your drug discovery program, but a pilot test would be really important before we could choose any of these modules for your programs.
Brooke, we have come to the end. Thank you, Elmar and Ivan. Thank you so much for sharing details about CellarisBio. We have a few minutes if you would like to take the questions from the audience. Are there any questions you'd be able to answer at this time?
Sure. Maybe I can start with a couple here. So Thomas asked the question, is there any way that this technology can confirm if your ligand is an agonist or an antagonist, I guess? We can't. So what the technology is designed for is only for direct target engagement. It's not a functional readout of whether it's an agonist or antagonist. We can only confirm whether this particular ligand binds to the target or not. So it's not a functional readout. So from your understanding, we can only say if it binds or not. That's absolutely correct. So it's only whether it binds or not. Another question was, Devin asked about, do you see differences in tagging the carboxy or amino terminus of the same protein? And we do. So MTH is an example where we tried both amino and carboxy terminus tags and tested it with binding to S-crizotinib, a well-characterized molecule that binds to MTH1. And we can only see binding of that molecule if we tag the carboxy terminus, but not the N-terminus. And it's well known that S-crizotinib binds to the amino terminal portion of the protein. So that where you place the tag does matter.
Ivan, would you like to answer the question about fluorescent-based tags?
Yeah, so this is a question by Sagar. What are CellarisBio strategies to overcome fluorescent-based tags from being degraded as form portion of the protein macromolecule by defense protease machinery. So we don't use a fluorescence-based tag. So the way our technology works is we use a FRET substrate. So our enzyme complementation creates an enzyme that is that will cleave this RNase. So it creates an RNase, a whole RNase that cleaves a FRET substrate. So once that FRET substrate is cleaved, that's when you get fluorescent signal. So it's not a fluorescent tag per se.
Right. There is a question from Kanav here asking about designing drugs for human ailments, but using preclinical studies would not be precise. Would it be better to use organ models in vitro and in silico? This is a great question. So in order to discover therapies, first of all, we need to know if your therapeutic modality is on target. So the target has to be defined, and then we have to find the right potent and selective therapeutic modality to it. And this cell target engagement system serves precisely for that purpose, right? So that you can be confident about your target drug pair. Once you have that confidence, now you can transition to using more sophisticated and more disease-relevant models, like your organ models, your patient-derived xenografts, and other, basically sophisticated models. But in the first place, that therapeutic modality has to be discovered and well defined. So this is why this is important, which serves you as a stepping stone to more sophisticated, more disease relevant models. And another question from Kanav is, can you elaborate on the AI integration? This is something we will have a future webinar dedicated to. So I would not want to tell too much about it now, except to say that, well, any drug discovery effort, especially using, large libraries, creates a vast amount of data. So, there is a positive data, there is a negative data, and that data itself is already a gold mine for future discovery efforts. You can train algorithms around that data, and then you can use it to discover better structures of your small molecules, of your ligands. And there are various ways you can actually optimize the medicinal properties of those drug candidates. So there are ways to do AI integration. It's the first step towards that is, what is your target, and do you have a good starting point? So a pilot test is really important for us to really start using the various applications of this platform. Are there any questions we want to answer? Brooke, are we good with time?
We're a little bit over, Elmar. Do you want to take another one, or shall we leave it for email and further contact? It's up
There's
to you.
one more question, it looks like, from Thomas. Have you any experience with multiple ligand binding to the target? In my case, some of our allosteric ligands need another ligand to be in the LBD to bind to our target. Can you see both events separately with this technology? So if there are two different binding events, we should be able to see both stabilizing the target. Now, one thing that we have come across is one particular target that has two different binding sites. One binding site results in stabilization of the target in a thermal challenge, and another binding site on that same target will result in destabilization of that particular target in the same thermal challenge. So we can pick up those two binding events because one is a stabilizing event and another is a destabilizing event. So it's a very interesting target that we've worked with, and it's because the different ligands bind to different regions of that particular protein. In your case, it looks like you have two ligands binding to different regions on the same target. Now, we should be able to see both events because you would get stabilization by one and an increased or enhanced stabilization by the other, or a change in the protein conformation or the conformation of that particular protein, which will impact its thermal stability.
Let me
Excellent.
answer.
Go ahead.
Yeah, I think this question is already answered by Deb. And is there any difference between C and N terminal tagging as a preference? I think, Ivan, you answered it well, that we're agnostic to where we want to tag the target protein. We would prefer by default to use the C terminus for that purpose. Well, thank you so much.
Yeah, excellent questions. Like Elmar mentioned, there will be future webinars, so please stay tuned for those. You can register on the CellarisBio.com website, and please feel free to message through email at hi@CellarisBio.com. Thank you to everyone for your great participation and for joining us today.